In this video, we will discuss about how the researchers had designed this mRNA for in vivo CAR-T cell generations. And we have discussed the important part of the CAR construct. That is, it should have a cellular domain, hinge, transmembrane domain, and the intracellular domain. So one of the example is shown here, where this SCFE is the cellular domain, and CD alpha is the hinge, and this CD 3 zeta is the intercellular domains, and CD28 is the transmembrane domain. So they use different combinations of the mRNA and also do the codon optimizations to find out which CAR construct give them better T cells activations. So this is some of the examples that are shown here, how they have did it. And in the panel B, we can see that they not only changed the codon optimization and the CAR construct, but they also tried different 5' UTR and 3' UTRs to find out which one gives them a better T cells activations. So this is how in general their roadmap that they used for mRNA design. That is, they use the codon optimizations and at the same time UTR optimizations. Then they took this human primary T cells and then use their mRNA for CD19 CAR expressions. After that, they check the T cells for the various activation state, and they use the several activations markers that we have discussed in one of our previous videos, that they can use these activation markers could be in the form of cytokines that are released, or in the form of its interactions with the targeted tumor cells. And thereafter, once they find out the mRNA design that gives the high T cells activations, then they use the in vivo validations in the humanized mice for CD19 CAR expressions and co-cultured with this null M9 tumor controls and primary B cells that have this CD19. B cells, for this they use the CD19, but when they use it in the mice, they targeted CD8 plus T cells. So let us now discuss the results that they have seen. So for this different kind of CAR construct, we can see that they're with the improved in the CAR functions, they can see the amount of this area of this null M6 tumor in the micrometer square per image decreases. And so they, from this results, they are able to find out the CAR that gives the maximum CAR that gives the minimum tumor areas, and then they use those CARs to check the in vitro results, so check the CAR expressions in the in vitro studies. So for first, they check the CAR expressions and compare it with the base CAR. They can see that the improved CAR has the more CAR expressions compared to this base CAR. And then they check the different cytokines when they co-cultured it with different cells that has CD19 or different targeted cells, for example null M6. And for that, they check the results are shown here for IFN gamma. You can see that the IFN gammas are overexpressed in the improved CAR. Similarly, for the Raji cells, they can see it is overexpressed, but for the K562 cells, they do not see overexpressions of the IFN gamma. But for these Raji cells and null M6, they can see a increase in the IFN gamma. In this case, they use the CAR expressions that can target CD19 that are present on the targeted cells. And now they also did the after getting their increase in the cytokine expressions such as this IFN gamma, they did the in vivo studies in the humanized mouse, where they target these CD8 plus T cells, where they use the CD8 plus targeted L829 targeted lipid nanoparticle towards this CD8 plus T cells, and they can see that compared to the base CAR2, their improved CAR2 has a high percentage CAR plus T cells expressions. So from these results, Researchers wanted to say that they have optimized this mRNA design using various strategies that I mentioned in this panel C to panel C's and the results that are shown in this whole figure to find out the optimum CAR mRNA designs that can give more CAR-T cells in vivo generations. That is shown in this panel G, that is done in the humanized mice. So that's all for this video. If you need to understand more about the mRNA designs for the in vivo CAR-T cell generations, you can go through the reference that has been provided in the description. Thank you for your time.